Okay, this is the second set of chapter 2 notes, or sorry, chapter 12 notes. I can't remember, some of this information may be in chapter 13 as well. We talked about briefly in the last video. I'm just going to go over a little bit more detail about reproduction in prokaryotic cells. So we're going to go fast. Remember that the DNA of a prokaryotic cell is usually a single strand. It's very simple. It replicates, unzips, copies, and you have two cells. They should be clones, identical, unless there's some kind of mutation. There can be some other ways of them getting recombination, which is just recombining their genes between different bacteria, but it's still an asexual type of reproduction. Come on. Yeah. So it's asexual reproduction. Means only one parent is needed. The DNA replicates, cell pinches can happen as quickly as every 20 minutes. Might stop dividing because they run out of food or space. Budding, we talked about in the last one too. It's very simple. Creates a bud, and then the bud breaks off. This is where I want to really elaborate, how bacteria can exchange genetic material. Because they can, even though they're asexually producers. One way is transformation. This is where you have a plasmid, and the the plasmid has an extra DNA that can be exchanged between bacteria. Transduction of a virus carries bacterial gene from one cell to another, picks one up in one cell and drops it off in another. Conjugation, two bacteria get close together and form a conjugation tube and exchange DNA. It's not a reproduction, okay? It's just a way to exchange DNA. And they do it to get more variation. Because remember, more variation increase means more survival. So these are just diagrams of the three different types. This is a picture of transduction, so there's some genetic material in here that would pass on and then maybe pick some up and then pass that on to the next cell. And then this is a conjugation tube here. They pass genetic materials down that. So there's really four ways that bacteria can get genetic variation. First of all, their DNA can mutate. It can just change, maybe because of an external environmental factors. Transformation is with plasmids, transduction is with viruses, and conjugation is with a conjugation tube where bacteria are close together. And that's it. Shortest video yet, so my next one.